Okay, thanks everybody. My name is Abby and I am the lab manager at the Aquatic Diagnostic Lab in Roger Williams University. And we mostly focus on shellfish, but today I'm going to be talking about our ability to detect pathogens in fish aquaculture and aquarium systems. So I worked with a number of collaborators that I just want to highlight, and most of what the story I'm going to tell focused on aquarium systems, but we want to be able to apply them to aquaculture finfish as well. So pathogens, there's a large number of pathogens, whether they be bacterial, eukaryotic, or viral in origin, that can enter into these systems. They're often resistant to any therapeutic agents. They can have a complex life cycle in which they have encysted stages. Those encysted stages then allow for periods of dormancy. So you will have sometimes weeks to months where you think you don't have any infections in your tanks, and then they end up having a disease resurgence later on. A lot of times facilities are going to focus on preventing the introduction of these pathogens using different treatments. But oftentimes, like I said, these pathogens will be resistant or be able to overcome those treatments. And right now, most of the methods that are for detection in these systems have to do with cytological examination. So after you see infections visible on the fish or see any abnormal behavior, that's when you tend to test and look for what's causing that. And this happens with skin scrapes, gill scrapes. These are very low sensitive tests, and it's trying to find a needle in a haystack. They're not the best tests out there. So our goal is that we need to have a more sensitive test, something that gives you an idea if there's any pathogens present in your system before you even see any visible signs on the fish themselves. So I'm going to talk about the pathogens we talked about, but we hope to adapt them to any others that might be important in some of your systems. So we worked with cryptocarrying irritants. This is the marine counterpart to the freshwater ick. That causes these very visible white spots on the fish. We had neobenedinia, which is a skin fluke, and these are larger eukaryotic organisms that are easier to see with the naked eye, and they tend to attach to the skin and the gills, as well as even the eyes. So in our case, we were able to detect 2 different species using our test. And then the ciliates. So ciliates tend to be a larger problem. They're cryptic species, meaning that a lot of them are morphologically similar. So when you look at them using a skin or gill scrape under the microscope, you might think it's one species when it could be another species. These are also free living, they're ubiquitous, they're likely present in your systems. Some of them become pathogenic at certain levels in the tank. So it's hard to track when infections may occur. Just going a little bit deeper, so crypto, like I said, causes these white spots. It's this trophont stage that embeds within the scales in the epidermis. This life cycle is complex. It has that cysted stage, so it can be resident in your systems even when you don't think anything's happening. And most of the treatments when it comes to any of these pathogens have to do with changes in water quality, temperature, salinity mostly, and then also chemical treatments. And for some chemical treatments, especially in aquarium systems where you have any number of different species in those systems, you have to remove elasmobranchs, for instance, out of the system before you can treat. So it's expensive, it's costly, and it's time consuming. Neobenedinia, they're larger organisms. They also have it's a direct life cycle, but there is a free swimming stage that we look to target. They have low host specificity, meaning that one species of neobenedinia can infect multiple different fish species, and that becomes important. And again, chemical treatments for these organisms. And the ciliates are the ones that are kind of our problem, or the problem we encountered. So we originally focused on uranema, and we thought that was the main issue in a lot of these aquarium systems. But when we were developing our diagnostic, we found that there are other ciliates that are culprits, and they tend to cause ulcers on the skin. These ulcers can go a lot deeper. It's also an open sore then on the fish, so you're now open to opportunistic bacteria or secondary infections. And you can just see here's two different species. Morphologically, they look very similar, so you can understand it's probably difficult to then identify them just using microscopy. So these are the three species that we are able to test for. Our goal was to design a molecular diagnostic, so we used a qPCR, and I'm not going to go into nitty-gritty details of that. Most people know, thanks to COVID-19, that we can test for a pathogen. We can has really high sensitivity, meaning before you might even show symptoms, and we can get an idea of how much of that pathogen was originally present in our sample. We then wanted to find a way that was easy sampling or passive sampling for aquaculturists and aquarists to be able to do. So that is a part of our eDNA. So environmental DNA means that we're going to look at and take a sample of anything and everything that's in the water, from your microscopic organisms to flakes of the larger fish, skin and something. We'll be able to detect that in the water. And then we want to validate these methods using known pathogen levels. Be able to track an infection in a tank so we can say, hey, we can detect it a couple weeks earlier than you see any visible macroscopic changes, or maybe even a month early. So I just want to talk about the life cycle a bit. So I'm just going to use crypto as our example. But most of what we're used to is seeing this trophont stage on the fish. I like to think of this as a trophy stage. It got the fish. It's happy on the host. And then you have this encysted stage, this tomot stage, which replicates and creates hundreds of tomites. And once that cyst bursts, these tomites enter the water column. So it's really this stage, this theront stage, that we are going to focus on being able to detect in the water. So you collect water, you're hoping that these, if they are present in your system, are in our sample, and we can identify how much is in that sample. Traditionally speaking, for eDNA efforts, we often used 1 liter water samples. And almost everyone's systems are a lot larger, and so this is a very small percentage of what we are trying to target. If you're only looking at 1 liter, even in triplicate, only 3 liters. You collect the water, you concentrate everything that's in that water down onto a filter using vacuum filtration. You can then extract the DNA and run a qPCR that's sensitive for those 3 pathogens I mentioned. We found when we started this work, we worked with one aquarium in particular that had a problem with crypto. So here we have salinity, and then a secondary x-axis that has or y-axis that has our number of individuals across a period of time. And so we found that using the 1 liter samples, we weren't being sensitive enough or detecting the infection at the right time. And so what you see here is they were seeing visually infections at low levels in yellow, medium levels in orange, and red levels were high infections. But what we got from our qPCR, which are these red bars, is we did get positive hits, which is good, but only after the peak infection. And so that's a lot later than we want for this assay. We would love to be able to detect organisms before we even get to a low visible infection in the fish. So to do that, we were able to improve on our sampling techniques using these Watera capsules. So these are larger capsules, have a lot of surface area. They're often used for like groundwater treatment plants. But it was very simple. Our collaborators were able to set it up, let's say like 4 p.m. one day, let it run overnight using a peristaltic pump and a pre-filter, and come back, collect the capsule at 9 a.m., so 18 hours, and we were able to increase our sampling 600 times and sample through 600 liters of water. So really being able to increase, hopefully, that sensitivity. I'm just going to go into some of our results. So we had some skin and gill scrapes, those original diagnostic methods that these facilities used. And we have our percent of samples on the y-axis and our different targets here on the x-axis. If you remember, we started with just the urinema. And so if we had concordance, meaning our visual diagnostics and our qPCR matched up, you're going to see a solid bar. Red is negative, both negative in visual, both negative, and negative in qPCR. Blue is positive. If we have discordance between those two methods, they're in the stripes, again, with the colors staying consistent. So what you see is we have a high concordance for both our targets for cryptocarrion irritants and those two species of Neobenedinia, right around 80%. 80 to 90%, excuse me. We didn't really have any positive samples for crypto, which is good. So there's no big issue there. But the problem here is that for Euro, we have a really high percentage of discordance, meaning that some of these facilities were seeing infections using skin or gill scrapes and identifying ciliates under the microscope that they thought were urinema, but in fact, the qPCR, we were not able to detect any urinema marinum. So when we redesigned and added in those other two species to our assay, we were able to decrease those false negatives where there was visual positives but qPCR negatives and increase our concordance. So we were able to confirm more positive results using the qPCR on these skin and gill scrapes. When we move on to our actual clinical samples, same idea here, colors are consistent, 3 different targets. We had 7 participating facilities, and most of them didn't have infections, which are great for them, bad for my study. But so most of the concordance was negative visually and qPCR. But again, when we go to adding in those other 2 targets for the scutocociliates, we increased our concordance, but now we've increased our sensitivity. So what I want to show here that's different from the last slide is that with these Wattera capsules, these blue and white stripes mean that we were having positive qPCR detection when they weren't seeing any infections in their fish, in their systems. So that's great. So basically, yes, we have a working qPCR. The eDNA system seems to work really well, those capsules, and being able to increase the volume that we're sampling. But we haven't gotten to validating our methodology, and that's one of our goals. So future work coming down the line, we really want to be able to inform aquaculturists and aquarists with like known numbers. So we get a qPCR value, not everyone understands that. We want to be able to relate that qPCR value back to some sort of estimated organism number per liter so that they can then treat properly. And then also, this is not real data, but basically what we want is to see our number of organisms, and we want to be able to detect that before we see, again, any low infections, any visible infections. And besides that, we're looking to expand on other pathogens. Maybe if there's a bacteria of interest you guys want to look at, or viruses, I'd love to develop primers for that and start looking into it. But yeah, so I'd like to thank my collaborators. I also really want to highlight Sandy. She was an undergrad student that did a lot of this research. So we're very fortunate that she was able to join us. And if you have any issues, please contact me. Thank you.
So we have time for maybe one question, one good one. Remember to repeat the question.
Yes.
Great.
So working in these big systems with dozens of different species of things, do you have issues with inhibition, or do you think you have issues with inhibition?
So the question is in the larger systems, do we have any issues with inhibition? Not to my knowledge. So I do most of my work is on molecular diagnostics, and so I haven't seen any issues here, and we got other positive controls that I worked on to make sure that that wasn't the case. And the fact that we are getting some detection in those capsules that have like large volumes of DNA is great, but we have ways to combat that if that issue does arise. But as of right now, no. Thank you.